Friends, welcome to the Wednesday colloquium. Jyotishman is not back yet, so I'm still standing in for him. It just so happens that these two talks were on my watch, happen to be biology talks. Our speaker today is our colleague from NCBS. It's a pleasure to host her. Uh, she'll talk to us about bacterial replication repair. Uh, to introduce her formally, I invite Amitesh Anand, who is closer to her research area. So he will do a better job than me.
Thanks, Manoj. Thanks, everyone. And so we have Professor Anjana Badrinarayanan from NCBS. She has done her PhD in biochemistry from University of Oxford and then spent about 4 years at MIT doing her postdoc work. The title was something different, not the postdoc. Okay. So towards the end of 2016, she started her lab at NCBS, and she's been there since then. It's an honor for me to introduce her because I'm really in awe with the kind of science she does, in general, the area of research and the way she tackles the problem in that area of research. I think despite being a diverse crowd, which doesn't look today, all of you will appreciate this term DNA and its form chromosome, and especially they being a carrier of genetic information. And even a tiny change in the genetic material can have huge repercussions. A tiny change in genetic material of us can result in genetic disorders. A tiny change in genetic material of viruses can result in the dreaded pathogens that we see or that we experience, we are still experiencing, and their variants. A tiny change in the organism of my interest and her interest, also bacteria, can result in strains which are totally resistant to antibiotics, rendering all our existing therapy ineffective. My lab personally is benefited by these changes because that's the raw material for the evolution that we use, but it's not good. No organism wants these changes to happen all the time. And that's why there's a very interesting repair mechanism operative in, if not all, most cells, to take care of these genetic damages that happen during the course of replication and etc. She looks into various mechanisms that are involved in these DNA damage repairs. And one thing that I really like about the research program in your lab is to look into the adaptive tailoring in those mechanisms. So how that co-evolution takes place to take care of emerging needs in repairing the genetic material. And I'm sure you are more interested in listening to her than I. So I'll invite Anjana for the colloquium talk. Thanks for being here.
Thank you. Thanks, Amitesh. Thanks, Manoj and Jyotishman for organizing this with Sandhya as well. Amitesh already gave the talk. So I'll just show you some pretty movies and then we can go to tea. No, but in all seriousness, I prepared something that's quite general to begin with and then gets into a lot of detail towards the end, right? And so because I was not so sure how broad the crowd is going to be, so for you biology aficionados for whom you think about this all the time, it's a revisit to the past to sort of position the question that we ask in the lab. And for those who don't think of biological problems often, I hope this is inspiring enough that you can appreciate then the types of science we do in my lab. All right, so what I'm going to talk to you today about is work that we do to track living machines inside bacterial cells. And we specifically focus on two processes inside these cells, the process of replication and repair. And as I go forward, I'll tell you about why we are particularly interested in these processes. But let's take a step back first and think about life itself. Now, these are some really spectacular imaging movies of cells growing and dividing. And across all domains of life, for life to propagate, a cell has to divide into two cells and ensure that its genetic material got carried into the progeny, got duplicated into the progeny with efficiency and with faith, which means that no deleterious changes occurred at the time of cell division. You know, Francois Jacob has said that the dream of every cell is to become two cells. And to do that successfully, cells need to be able to not just ensure that they divide, but also make sure that their genetic material is propagated into the daughter cells at the time of division. Now, what is this genetic material? This is an electron micrograph image of an E. coli cell that has been lysed out, and the DNA is outside in gray. This DNA is compacted nearly 1,000-fold to fit inside a living cell. And inside this living cell, this DNA is faithfully replicated, it's duplicated. So you're seeing here a cell in black and the DNA inside in gray. This in green, this DNA is duplicated and then it's divided and given to the daughter cell. And this goes on for millions and millions of years. For example, in bacteria, and without so much accumulation of mutations or deleterious problems on the DNA. Now, replication itself, people have drawn this really nice, this is a cartoonic representation of how a machinery replicates or duplicates the DNA. And it looks very smooth. It looks like it's a well-oiled machine. Bits of precursors come in, they get added to the DNA, and then you get a duplicated unit getting extruded out from the replication machine. And one would think that this is how replication and DNA propagation occurs inside cells. And for the longest time, nobody was willing to even consider that DNA itself is a highly unstable molecule. It's hard to imagine that something that's so important for life is subject to damage. But life itself is the biggest trigger of damage. So as soon as metabolism kicks into a living cell, it creates molecules that will damage the DNA. Apart from that, across the lifetime of an organism, cells will face damage both from extrinsic mechanisms such as radiation damage, but also chemicals that are going to physically hit the DNA and cause various types of chemical modifications to these bases. Some of these modifications can result in mutations. Some modifications can result in physical breakage of the DNA itself. Now, in those conditions, cells no longer have that really happy propagation that I showed you in the previous movies. You instead get these monsters growing where cells will block division because they cannot divide as the DNA itself is damaged. They block division to make sure that the DNA can be repaired, and only after that will they resume the process of the propagation of life. And so cells have to do three important things when they face DNA damage. First, they need to respond to damage. They need to know that there is a problem. They then need to choose the right pathway for repairing that damage because every chemical modification is unique, and the type of pathway that can fix that modification also becomes unique. And finally, they need to make sure repair itself is regulated so that repair doesn't make changes that are going to cause mutations that Amitesh likes, but mostly life does not like, right? And so it is super important that all these three steps happen accurately during the course of cell division. Now, what's really interesting is that repair itself is the biggest source of mutation and evolution. And this is via a process called stress-induced mutagenesis, a process where cells will turn on the ability to mutate their DNA when they face stress. And so you create this really nice conundrum. When you have damage, cells must fix the damage. That is important. But cells have the ability to pick a pathway that is going to give them mutations, but also a pathway that is going to result in no mutagenesis at all. And this balance between choosing an error-prone pathway and an error-free pathway can allow cells to do genome evolution and adapt to new environments. But at the same time, maintaining this balance is important for survival, as too much mutagenesis is also deleterious for the cell. And so we are really interested in my lab in understanding how individual pathways of repair are carried out inside the cell, and finally, how this balance may be maintained to allow for genome diversification, but at the same time, cell survival. And we ask these questions in a very fascinating group of organisms, which are bacteria. Bacteria are one of the largest, most abundant living organisms on this planet. And they have huge diversity in not just their morphologies, but even in their genetic makeup. I think we seldom appreciate how diverse these are. Two commonly used bacteria, E. coli and Bacillus, in many bacterial labs. They're more farther related in evolutionary timescales than a mouse is from a human, right? So that's the amount of diversity we are sampling when we study bacterial systems. And one of the things that DNA repair does is it contributes to the ability for this diversification to occur and for bacteria to be one of the most successful colonizers on this planet, colonizing almost every possible niche that you can find on Earth. Right, so as I told you, in my lab, we are really interested in understanding how DNA repair occurs inside cells and how this process is regulated. And how do we do this? We take an in vivo approach. We like to look at things in their natural settings, so to say. I mean, it's still a lab, so it's not truly natural, but we like to look at the molecules in their natural settings, and we do this via microscopy. I showed these two pictures here because, this is the first time-lapse movie of a bacteria growing. This has been taken in dark field with the bacteria directly on 35 negative film. Guess the year. This is 1910. It's the first ever time lapse of this living organism. It's Vibrio cholera, the same organism Gandhi is apparently seeing under this microscope because it was an epidemic in India at that time. And this movie was taken at the Bombay Institute, which I guess doesn't exist anymore. Is it? Hufkins. Okay, thank you. Yeah, so it was taken there. So there's a long history of using microscopy to look at microbes. And what we do, we've come a long way from just simply seeing these microbes in this format. We've been able to now look at, yeah, and they did dark field. Yeah, and they did dark field imaging on it. And yeah, exactly. It's quite stunning to be able to have done that.
What do you mean by dark field imaging?
So instead of using light to create phase difference, they use the phase difference that's coming from the organism itself. So the light scattering from the organism, they collect that. So the imaging mechanism is the opposite of how phase contrast imaging would work, where you use light scattering to capture emptiness. Here you use light scattering that's coming from within cells or within any entity. It was used originally for rocks and so on, and then they started using it for organisms. Right. So how do we do this? How do we study these processes? We use, as I said, a lot of microscopy, but then we also use genetics to perturb the system. Join again, okay? Joined. I am connected on pass.
0407-2020.
Sorry, you guys. Okay. right? I mean, it's a bit moot if they can't hear me. Anyway, so I guess you can't see my screen, so sorry about that. But we're going to talk about this process called double-strand break repair. And it's a type of damage that occurs when both strands of the DNA get broken, which is shown here. So you have an intact DNA strand. And when your both strands of the DNA get broken, it's a double-strand break. It's one of the most lethal forms of DNA damage. If it's unrepaired, it results in genomic rearrangements, loss of genetic information, and eventually cell death. Now, across all domains of life, all the way from bacteria to plants and mammals, a faithful way, without making any errors at all, of repairing this type of damage is via homologous recombination, which requires an uncut copy of duplex DNA, which is an exact match to the above strand that's used as a template for repair. Now, this process has been studied across several decades now, and we know the key steps that go on to allow for this type of recombination to occur inside cells. Now, the first step in this process is called end resection, where the double-stranded DNA ends are chewed up by an enzyme, which reveals these single-stranded DNA overhangs. Now, these overhangs are then bound by a protein, and the identity of the protein will get clear in a moment. And this protein forms a filament on the single-stranded DNA end. And then the protein filament, the nucleoprotein filament, searches for a match, an exact match of sequence on the double-stranded DNA, the intact double-stranded DNA. And when it finds a match, it does a process of strand invasion, where it opens up that second copy and then uses this as a template to repair the damage. Now, the molecular details of how this search itself occurs are also really quite well characterized. And the study came out just a couple of years ago, where they were able to show how the base matching occurs on this nucleoprotein filament. And I won't get into the details, but if you are interested, I can come back here and take you through it. Right, so what is this nucleoprotein filament, and what is this protein that's binding the DNA ends? The protein is called REC-A, and it's one of the two proteins you'll have to remember for this talk. It has homologs in eukaryotes also.
Why did DNA get damaged? Is it mutation or?
DNA gets damaged from everything. So, for example, metabolism. If your cell is creating ATP, making energy, a byproduct of energy generation is reactive oxygen species. And these reactive oxygen species have a propensity to go and attack DNA and make modifications on it. So that's one source. Radiation will create also damage. Chemicals create damage. There's also mechanical sources of damage and so on. So there's many reasons for DNA damage, and it's constant. So there are studies that estimate that in a single human cell, your chromosome entities go through 10,000 damages per cell per day. And so this is in humans. So I'm guessing similar numbers are applicable to other organisms also. Some are more subject to damage than others because of the environment they live in. But damage is, part of life. It's inevitable, I would say. Yeah.
Base mutation rate across organisms, is it the same or is it different?
So it's vastly different. I cannot actually say what it is in humans. I do know in bacteria it's about 10 to the minus 9 per generation. I'm guessing it's around that range. So 10 to the minus 6 to minus 9 per generation. Yeah.
Is it in higher, like if you look at C. elegans and other things, they must have leaves over there. There, the DNA is folded around chromatin and histones. Does that protect?
No, so actually that's the thing, right? Most damage comes from intrinsic sources. So replication is one of the largest sources of damage. And so the minute you're replicating DNA, there's an error rate of your polymerase. That error rate alone gives you this number. And so this is baseline mutations without including these extrinsic factors. So just from replication alone, these are the rates. So then you have...
What is the spontaneous rate at which you get mutation?
So if it's replicating, then this is the rate.
Yeah.
If it's non-replicating, then...
Others.
Absolutely. So this is like replicating lab conditions. So you do not face any other stress. In replicating lab conditions, you get this as the rate. And then you add more to it as you have DNA stress. Yeah.
The eukaryotic replication machinery is said to be more efficient than the prokaryotic one. So wouldn't that reduce the?
I had no idea about that.
I've heard that like the DNA, I think gamma or DNA called gamma. DNA called gamma is more effective than DNA called, which is in the prokaryotes. That is counterpart in prokaryotes. It's more better. It's better at matching the bases and preventing.
Yeah, but it's all about what type of modification there is. So you employ an error-prone polymerase based on how much, what type of bulky adduct there is. Gamma is not always used. You also have pol theta and so on. So based on the type of modification, you may have actually more or less error. For example, the error-prone polymerases that work in mitochondria tend to be far more error-prone than the nuclear genome. So I'm not entirely sure one can say this. I don't know if one machinery the other. Yeah. Uh, elephants have low rates of DNA damage associated mutations, which is why they don't develop cancers so much. We do not know why. People think that it's about P53, this signaling molecule that triggers a bunch of DNA repair mechanisms also, but we don't know. It's possible. There are many organisms that have lower rates of mutagenesis. Corals are another example. So corals, they have very low rates of mutagenesis. These are harder to study organisms, so it's very hard to know molecularly why it is the case. But yeah, it's true, there are such organisms also.
RNA bacteria this happens much faster because it's even less, RNA is even less stable than DNA.
RNA bacteria.
The kind of things which, sorry, RNA virus, it's not bacteria.
Well, I mean, so in all of them, when they're replicating, they are going to then convert to DNA and replicate. And so when viruses are going to replicate, they're still going to come into the host and use most of the host machinery. So I'm guessing the rates are going to be similar as host machinery, but I don't know.
So as I said, Anjana's area of research is extremely exciting and fascinating. So I know that there will be lots of questions, but for another 15-20 minutes, if you can restrict the questions more towards what is being talked, then and after that, C will be around, so we can talk a lot more general questions.
Oh yeah.
Unless you're okay with them.
Okay, either ways, but maybe I don't know. Should I get to some data?
Go ahead.
Should I go ahead? Okay, I'll go ahead. It'll take me a while to get to data though, because I have to still build the question. You still don't know what the question is. So the question is starting here now, actually. So this process of searching, right? It looks quite straightforward when you draw these stick diagrams. But I remind you how the cell is. I showed you this DNA that is highly compacted in a confined space. This DNA is also occupied by a lot of proteins that bind the DNA and carry out transactions as well as organize the genome. And so in this very, very tight space, this nucleoprotein filament has to sample all of this DNA and find that exact match. Now, there's only 4 bases that make DNA in various combinations, so it's quite a conundrum how the right match is always found. And so I give you the example in case of bacteria, an average break may be approximately, the break end in vitro people have studied is approximately 30 bases or so. And it makes the right match if you use a 30 base long DNA that's searching, and the total amount of genome space that has to be searched is 9 megabases. So it's the match has to be right, and the match has to be found quickly. So again, in bacteria, this match is found within an order of few minutes, anywhere between 5 to 30 minutes. Now, if you do theoretical estimates of how long this can take, it will take a few hours for successful match to be found if you were to go through this sampling process, looking at every base inside the genome. Yes?
So if it is 30 base pairs over which you're looking for a match, that kind of sets an upper limit on how long like something can be, right? Because
let me correct that. So it's 30 bases total single stranded DNA. It's 8 bases to get to commit to a match.
Yeah, but whatever it is, as long as it's finite, it means that if an actual gene is longer than that, you have a probability of getting a wrong match.
Yes,
Yes.
yes, exactly. That's the conundrum. You have a high chance of getting a wrong match, but you do not get a wrong match. You get a right match. And we don't know how exactly that right match gets found so quickly in this space.
I meant wrong match, so it's like four alphabets. You know, you have these 8 over which you're matching, and then you have, let's say, the remaining things in that strand are unique. So you have two different genes which are same on these 8 but different.
Yeah.
And then break happens in one, it's supposed to repair over that, but your matching mechanism finds a match on a shorter side and so that would be a wrong match.
That is a wrong match, but it doesn't do that.
Then it's not searching over a finite space.
Why do you say that?
I mean, you said it's over 8 base pairs.
It's searching over 9 megabases. It's committing to a match when there's an 8 base match. So you search the whole room, but you stop when you find 3/4 of you. But you're searching the whole room. So the space is the entire room and every individual in this room. So it's not searching only that gene or only those 8 bases or only those 30 bases. It's doing a combination of, so it's using every nuclear, okay, let's get into the details. Every protein molecule here can bind at any given time 3 bases. Okay, so each one protein makes contact with 3 bases on the DNA. And now you have, so there's 3 bases speaking to this one, there's 3 bases speaking to this one, and so on. And each one is independently trying to make a match of its, and each protein itself on the DNA end, on the single-stranded DNA end, is bound to 3 bases, right? So it's almost a base-to-base match for 3 bases from this protein, a base-to-base match for 3 bases from this protein, and so on. Now, if you make a single base match, it's insufficient to stabilize this filament. You need to make an 8 base match. At that point, the filament will stop searching. But as long as you don't find 8 bases, say you find only 3 base match, the filament has a chance to leave this system and go back up there and go to a new position on that DNA and then do the same sampling process. All right. Okay, so right, so you do have to do the search process inside a rather confined space and across 9 megabases of DNA in the case of bacteria, but a lot longer in the case of human cells. So how do you do it? So again, because of the problem of time, the efficiency of searching is quite good, as one has seen already. I mean, 8 bases is sufficient. We do PCRs with shorter 20 nucleotide oligos and it works just fine. So overall, we know that this is not going to be a tough problem to find the right match, but it's going to be a tough problem to do it quickly. And so that's where the problem comes. How do you do it quickly? How do you make sure you do it not in a few hours? And so people have proposed various types of models to explain this. They've suggested that the protein filament can do some sort of jumping and hopping across different segments of DNA to speed up the sampling process. The protein filament can also do some form of hopping across these segments and then also 1D sliding, all of which may help it to move across the genome. But this will not give any directionality to the search process, and so it's fairly random where it samples, and it may end up sampling the same region many times inside the cell. And so it still doesn't solve this problem. It doesn't solve the problem of how inside a living cell, a break site that may be located here can find a homologous template located here, the distance between the two being approximately 3 to 4 micrometers, and as I already said, the sequence space being approximately 9 megabases. People have also tried to tackle it in a different way. Then they said, okay, it's complicated, may not work. So let's not think that the search happens across the entire cell. The search may happen in a confined space. So you do proximity guided search where you search only in a particular location around the break site. This is true in case of human cells or mammalian cells that are undergoing meiosis, for example. Search happens only in regions where the paired chromosomes may be there, and you don't do long distance search. But more recently, there are studies that are breaking this idea. First of all, we have to take into account the fact that in many bacteria, chromosomes are not held together and they go apart rapidly. Second, double-strand breaks, this type of break can occur outside the context of chromosome pairing. They can occur in cells that are not replicating also, for example. And finally, 3 studies over the past decades have shown that repair can occur across long distances inside the cell. First study showing this in E. coli, another one showing this in another bacteria, and a final study showing this again in 2 other bacterial species, which then suggests that recombination between these distant regions inside the cell is possible in bacteria, as well as in yeast more recently. And so there must be a mechanism that allows for a search that may be moving this break site towards the homologous template. And so that's the question we set out to ask in the lab a few years ago now. We wanted to understand how this nucleoprotein filament of REC-A finds a homologous repair template inside the cell. And this is the work of 3 graduate students in the lab. The first part of the work was led by Afroz, and then it's been followed up by Saurav and Inchira. And so the work we do is using Kolkata crescentus, which is our model system of choice. And I want to very quickly tell you about why this is a good system for us to study what we are interested in. Kolkata has a very specific life cycle. It's born as a single cell with 1 chromosome. It differentiates into a stalked cell and then initiates replication. And then at some point, it reaches this stage where it has 2 completely replicated and segregated chromosomes. And then it divides asymmetrically to give you a swarmer cell with 1 chromosome and a stalked cell that starts replicating already. And so we can use a genetic trick as well as simple gradient centrifugation to isolate just these swarmer cells with 1 chromosome or these pre-divisional cells with 2 chromosomes. Cells with 1 chromosome cannot do recombination because there's no template. And so in these cells, we can ask about search dynamics. And in these cells, where there are 2 fully segregated chromosomes, we can ask about long distance search. If you have a break at one end and you have a homologous partner at the other end, how do cells search for that partner? And so how do we make a break? So we make a break via genetically engineering a break site onto the chromosome itself. So what we've done is we've engineered a cutting site. It's recognized by a restriction enzyme. That site specifically cuts the DNA and makes a double-strand break. The site is located at the pole in the Colobacter cell, and it's marked with a fluorescent marker. And so we can now track, using this system, all the steps of recombination. So first, I'll take you through the red markers. So these 2 markers are telling you where your break site is and where your homologous template is. Now, at some point, when a break is induced, you lose your break site marker. So there's no more red marker here. And that's because of this chewing up that I told you about earlier. And at some point, towards the end of the movie, you see the red marker emerge again. That tells you repair has been completed. Now, on the bottom, we can mark REC-A, and we can ask where it is. And REC-A localizes at the point where the break has occurred. So you see it here. And then it forms some sort of a filament, goes to the opposite cell pole where this other marker is there, where the homologous template is there. And at some point, you get diffuse localization again, which to us is indicative of the fact that the REC-A filament has successfully found a match and has now dissociated from the DNA. Right? So now we can focus on this particular aspect of the process of homology search, and ask how is search occurring inside cells. And so to do that, we do time-lapse microscopy and we track the REC-A filament inside the cell after a single break has occurred. Yes? Okay. So we use a cell division inhibitor called cephalexin that blocks the final step of septum formation. And so we don't have cell division anymore. And we've also blocked replication initiation, so you don't get new rounds of replication. And so every time you see the emergence of the second marker, it has to have come from repair only, which is why this makes it a really powerful system, because there is no way replication could have given you the emergence of that marker. Right, so then we track REC-A inside the cells, and we observe that REC-A has a rather dynamic behavior. It has these corkscrew-like motions at the break site, and that over time then it starts to move. And so you can start to see displacement of the REC-A filament from the break site, and at longer time scales you can start to actually track it inside the cell and see that it moves from one pole to the other. It moves from where the break occurred at one cell pole, all the way to the opposite cell pole. And it also goes through these back and forth motions. So for example, in this chymograph, you see the Recce intensities at one pole, it goes to the opposite cell pole, reverses, and then comes back and goes through these back and forth motions. It sometimes is super fast, but on average takes approximately 300 odd seconds to hit the opposite cell pole for a cell that's approximately 4 to 6 microns in length. And it goes through 6 to 8 such back and forth motions before it finally finds the partner at the opposite cell pole. Now, the first question we had to ask was, is the homologous sequence sort of guiding this search motion? The answer is no. You can see this happen even in cells with just one chromosome where there is no repair template. And in this case, Recce just keeps going back and forth and doesn't stop at all, which suggests that the only way to stop it is by finding that right partner. And there is no backtracking. So we tend to not see Recce turn back earlier. It tends to turn only once it has hit the opposite cell pole. And we do think that the turning is based on the barrier. So it has to hit the opposite pole and that curvature before turning. And then we do see pausing. So along the way, it does take pausing. And so you would see stopping, the stop and go motion as it's moving. And so then the question becomes, how does Recce find the homologous partner, right? So how do you get a filament like this, which we've known for a while now, that such a filament must likely form inside the cell. But we didn't anticipate that this filament would be so dynamic and be so mobile. And so how do you get this filament to move? And then how then do you get finally recombination? And we know that this motion is required for recombination because without this motion, you don't get long distance repair. And so that's the question. And so going forward, I'm going to try and answer these questions for you, although we don't have so much time. So I'm going to see how far we get. I'm going to try to get to this question faster because that's where the fun stuff lies. But I'm going to quickly tell you whether the location of the break site matters. Does it matter where you cut? I'm going to tell you whether the chromosome density itself acts as a barrier to Recce motion. I'm going to tell you where the two break ends are. I don't know if any of you wondered, there's two break ends. Where are they inside the Recce filament? I'm going to tell you about that. And finally, I'm going to give you some hints as to how this filament actually moves. Right, so very, very quickly, we wanted to ask, how does the location of the break site impact this search process? And so for this, what we did was we programmed break sites. So the first break site I showed you is here at the opposite cell poles. We programmed break sites going all the way from 1 o'clock position, 2 o'clock position, to 3 o'clock position. Is that 3 o'clock? Yeah, 3 o'clock position inside the cell. And so now you get this distance that reduces. So between these two positions, your distance is going to be lesser than between these two positions and so on. And I'll show you a graph later on about how these distances look. And we have a trick to also label these break sites, and I can tell you about it later. So what we can do is in all of these cases, we can track repair. So you have 2 dots where there is no break. When there's a break, you get a dot. And when there's repair, you get 2 dots again. Right? And so it's a very simple assay now. And then we can just measure repair time. Right? How long does it take from when you lose your break site to when you gain it again? And as I told you, the distances do change quite a bit. So the distance, say, for example, between, so if we say the origin is the longest distance, you see this nice reduction in distances as you go closer and closer, like the 3 o'clock mark. And I will show you data about what happens if you make a break right where the template must be next to each other. And so what we find is that in all cases, overall repair occurs quite rapidly. It occurs within a generation of cell growth. So it's before cells need to divide, but there is a lot of variability. Some markers seem to repair faster than other markers, and we don't know where this difference is coming from. And we also don't know if this difference makes any difference to cell growth itself. But we do note that this particular marker, these 2 markers, do have a faster repair time. And it's exciting because these 2 markers are next to highly transcribing regions. And so we do think that maybe having open genomes allows for faster repair in this case. And just to let you know that in all cases, we made sure that the cell lengths and volumes are comparable so that when we do calculate repair time, it is coming from a difference truly in the repair time itself. We also tracked then search time. So we did this for Rec-A across all these break sites as well, and asked how long does Rec-A take to find the partner, and if Rec-A does the same back and forth traversal in all these cases. And so we did calculate search time, and then we calculated the traversal times also. And it's super interesting to first note that Rec-A does move no matter where the break site is. It'll move even if you have a break at 3 o'clock position. And in this case, it can even overshoot the marker here, go all the way to the opposite cell pole and do the back and forth motion again. And the search times do reduce concomitant with the repair times. So Rec-A is able to find the partner faster next to the highly transcribed regions, but it takes longer even if the distance between the template for repair and the break site itself are short, right? So this is telling us that there's something about the genome itself that may be dictating the search times. And it's not just about physical proximity of the break site and the repair partner. This feeds in really nicely to this final experiment we did in this context, which is quite curious. So we thought, okay, if you have a template that's right next to the break site. So now in this case, we make a break here and the repair template is within 300 nanometers of the break site. Here, would you get movement at all? And could it just be a case of first encounter? The filament forms and then it encounters and then you do repair and it's successful. And so we tracked Rec-A in this case, and we observed two kinds of motions. We observed a motion where Rec-A moves, and it does search, and it shoots past what should be the repair template, goes all the way to the opposite cell pole, and does the back and forth again. And in that case, the search time is quite comparable to the other markers, because now it has left its point where it started, and then it's all about the chromosome organization alone. But then you do have this search time that's super short, when there is no movement at all. So there is a chance of first encounter. Filament forms and finds the partner, but it's possible that if it doesn't find a partner within a window, it takes off. The number of times we see no movement is only in 20% of the cells. So in most cells, Rec-A commits to traversing. It commits to traveling to find the partner, and in only rare cases does the first encounter alone suffice to do repair. This suggests that the program of movement is sort of encoded in the filament and whatever else is regulating the movement itself. Yes. Yeah, okay. Yeah, so we've been very curious about that. Does it pause at similar locations? We've done all these analyses to see whether pausing locations inside the cells are similar no matter where the break occurs. There doesn't seem to be anything that pops out as a specific location where it will stop. Pause frequencies are also variant based on the length of the filament, but the time it takes overall to search seems to be conserved. So I think there's two properties here. There's something about the movement that is independent of the intrinsic property of the protein. And then there's the property of the protein itself, which is this 3-base searching. And that the protein seems to hold. But the movement itself, I think, is extrinsic. And that's why you don't see any of these patterns of does it stop at the same point and so on. And I'll come to the molecule that drives the motion, and maybe that can explain why is it that there's no pattern in the movement.
And then we have said, how fast does it move?
I am going to get to it. So yeah, how fast does it move? It's about 5 to 6 microns in 300 seconds. I have a velocity graph somewhere. Let me just show you that. How many molecule lengths? Oh. Okay, so here's the problem, right? I mean, the analysis is a bit confounded because it's hard to measure the movement of an entire filament. So we have to choose a point. So we choose either the centroid, which for the most part tells us the movement, but then we've also done this for the leading and lagging edge of the filament. I'll get to the numbers for you. Sorry?
Imagine that it is somebody who's sitting in a
Yeah. It's fast, but it's motor fast, like it's at speed of motors. So yeah. All right, so does the location of the break site matter? Location seems to have some effect. An example is the location right next to the repair template. In that case, you get this likely first encounter-based repair. And then the other example is the part where there is high transcription. That also seems to speed up repair. But whatever it is, repair doesn't take longer than a single generation time of bacterial growth. Okay. Does chromosome density affect Recce mobility? Because we are short on time, I will give you the answer. Yes, chromosome density does affect the path of Recce. So it is seeming to move through the chromosome. And we think that what it's doing is it's taking this. So we tested these three hypotheses. We tested the hypothesis, does it cut through the chromosome? Does it go on the surface of the cell? Or does it do the zigzag motion? We used turf microscopy to image just the surface of the cell to ask whether we can capture a filament moving at the surface. But what we find is that our data are consistent with the zigzag motion. So it's coming in and out of the surface of the chromosome. So it's sort of going in, out, in, out, in, out. And the density has an effect on this in-out motion. So when you have less dense cells with chromosome, you tend to have a faster motion through the chromosome because there's no likely impediment. But then when you have a super dense cell with more chromosomes, you have more of this in-out motion with several times when the Recce filament samples the surface also. Right, so chromosome density does affect the Recce path. Right, so answer the curiosity question, where are break ends? If you think about it, there are two break ends made here, two filaments that should have loaded on both those break ends, but only one filament seen. We can do this for many breaks also. So we've made monster cells where we can make, 3, 4, 5 breaks. And in all cases also, you'll see many filaments loading and then they all fuse together into one single filament. And I will tell you later what this is not dependent on. So overall, break ends are within that one filament that you're seeing. So two ends are moving together, right? So now we can have a picture. I'm very proud of this animation that I'm not very good at art. So this is the level of art I can do. Here's an animation of what we think is going on with the filament. You have two break ends, two filaments load, they fuse into a single filament. And now this filament sort of moves around in the cell, turns at poles and comes back. If you note, I show that a bit of the double-stranded DNA is also moving with the filament. This comes from data. We have tracked double-stranded DNA next to the break site, and we can see motion of that DNA also with the Recce filament. Right, so in the last few minutes, I'm going to try and answer to you how this filament moves inside the cell. Because Recce alone has no motor property. It has ATPase activity, but yes.
Induce a break at a particular location. In your image, but there should be other breaks taking place as well.
No.
None at all.
No.
You're completely.
Yeah, it's a super clean system. So this enzyme is coming from yeast mitochondria.
Just about natural causes, no? There's lack and stuff like that.
Well, I mean, so at least in our case, if you saw more breaks occurring because of phototoxicity, for example, you'd see new filaments loading at that site. We don't tend to see that. So you tend to load a filament only where the break occurred, and then the filament dissolves when the break has been repaired. So at least in the...
Didn't you have more?
Yes, absolutely.
So in that case, how do you do the specificity?
I'm so glad you asked this. This is why we made this system. We're still playing with it. So here we make multiple breaks inside the cell. And we wanted to ask whether you have parallelized search. So you have many filaments searching. What we end up finding is that you actually, cells sort of, the Recce almost coalesces into a single filament. And that filament then does search. So it's possible that what Recce is holding inside it is multiple break ends. And in that motion, multiple break ends can multiplex search. And this has been seen in vitro also. So if you may build a RAD51 or Recce filament, you can multiplex search within a single filament.
They separate when they find?
Most likely, we don't know. We don't know. And I mean, I'll jump the gun here. I want to show this very quickly. These data. So they came out late last year. This is in yeast where they make 4 clustered breaks and they look at the RAD51 filament dynamics. They see exactly what we see. A filament building on these breaks in yeast, this RAD51 filament sort of encompassing 4 breaks together, and then you get this motion. And they build a model to explain how this motion can make search more optimized and reduce errors. Their hypothesis is that it reduces errors. Now, I don't think we know or they know how is it that you've organized all these breaks into one filament.
I do not understand why it precludes the ability to form a second filament.
No, it does form a second filament, and then it coalesces. So the point is you do make multiple filaments, but then you coalesce all the filaments into one. I don't want to use the word, but it may be, yeah. So there's this coalescing of multiple filaments happening, and we don't know how. And it seems to hold in eukaryotic systems also, which is what is mind-boggling. All right, let's quickly end the mystery here. The idea, so we don't know. So the idea is that you have multiple break filaments loading at multiple break sites, and when you have motion, they coalesce. And once they've coalesced, they don't separate. So then you see only one filament moving inside the cell. And so we are assuming that multiple breaks are within the filament, but we don't have the resolution to see the break ends themselves. And so this is why we don't know where really the break ends are. Yeah, so I mean, I'll come to this in a moment. So there is this one, where is this mystery protein? Let's come to that. Okay, let me just tell you this answer, and then maybe that. No. Okay, I think maybe telling you about this may answer a bit more this question, right? Okay, so the question is, how does it move? And so we went searching for potential partners, and we decided to focus on a class of proteins called the structural maintenance of chromosome family proteins. And the original reason why we focused on them was because we thought something must hold the break ends together. And these class of proteins are known to bridge DNA. They're also known to have motor activity and organize DNA inside all living systems, right? So there's many types of these SMC proteins. There's one in bacteria, highly conserved, almost as conserved as REC-A itself, which is quite conserved. And it's called REC-N, and it's the second protein we have to remember. We know that REC-N interacts physically with REC-A. REC-N has an effect on REC-A function in vitro and is essential for doing double-strand break repair from really old data that originally found this protein in E. coli. We also know, as I told you, this physical interaction and this essentiality for double-strand break repair specifically. Right, and so obviously the question was to ask, what happens if you remove it? And it's very dramatic. There's no motion. There's zero movement when this protein is absent from the cells. You can see this across, a really long time scale now. This is imaging done up to 120 minutes. You see a filament build at the break site, but there is just no motion of this filament at all. And so just to remind you again, this is what the wild type curve looked of position across time inside the cell for the centroid of the REC-A filament. And then in the absence of REC-N, it just is not mobile at all. And I also want to remind you that without REC-N, you get no repair at all. And this is immaterial of where we make the break. It's not just about making the break at the pole. We can make the break anywhere inside the cell. And if you don't get a mobile filament, you don't get repair from REC-A. And so we know now that REC-N is absolutely required to drive this motion of REC-A. And so the question obviously then becomes, how is REC-N driving this movement? But we are at 4:59. And so the question becomes, 5 minutes. Okay, in 5 minutes, in 10 minutes. Okay, in 10 minutes, I will try to answer you very quickly what we think is going on. We have a working model. Number one, REC-N is not holding the breaks together. So even if you don't have REC-N, you still get a single filament only. You can even capture the merging events. So we can do fast imaging to see the two ends of the break loading, and then they merge together. We can see this for both these wild type and REC-N deletions. The times are similar also. So it's not holding the filament together, but we notice that apart from movement getting affected, one other thing gets affected also. The Recce filament, if you remember, I showed you, has a lot of dynamic shapes when it's moving. Rarely do we see two, but you see these bent filaments and so on. In the absence of Recce, this dynamicity is lost. And so it doesn't work. Anyways, there's a movie here of Recce, which actually just looks like this for the entire time series. It's just a filament that's static. It's stiff. And if you calculate the fluctuations in length, you lose these length fluctuations in the absence of Recce. And then if you just simply do some mapping of the number of vectors required to fit a filament, if it's straight, you get 1, and that's what we see for Recce. It's just this really stiff filament that doesn't seem to bend at all. While in the presence of Recce, you get these really nice bend motions and this filament length fluctuation. We can see this even more clearly with 3D imaging. And so, in a wild type cell, you get these nice long filaments that span across the cell, and consistent with our TIFR data, you can see it sometimes at the surface also. For example, here you see it go across the cell. Now, what's striking in Recce deletion is that you see the stiff filament again. But when you look in 3D, you realize that the filament is actually bent over each other and super stiff. So it's a rod that's just stuck. And it has no angles associated with it. And so it appears that Recce is inducing some kind of bending in the filament, and this bending is associated clearly with mobility, because in the absence of Recce, you lose both bending and mobility. So we then decided to ask, okay, can we track Recce and see where it is inside the cell? And so we tagged both Recce and Recce inside the cell, and we first asked where it is. Skip this part. It appears asymmetrically located on the filament surface. But what we found really striking is that it's super dynamic. So Recce forms this very nice stable filament, and then it starts its search process and remains bound for a few minutes until search is complete. Recce, on the other hand, has fast kinetics. It's binding, unbinding super rapidly. We don't detect it for more than 20 to 30 seconds per binding event. And it's mostly binding as a fluorescent focus, so it's not elongated. It's a dot that's binding and unbinding. And so we can try to track these motions across several cells and ask, what type of dynamics is this revealing for us about Recce? And so we see 3 types of localizations in any given search movie. You see non-homogeneous Recce, so a cloud of Recce molecules around the filament. Then you see these transient localizations that are short-lived, and then you see these stable localizations that end up towards the lagging edge of the Recce filament. And so we can do analyses of these and extract from it intensities as well as binding times. And I will not go into these data, but I just want you to remember these 2 things, that there are these transient localizations of Recce binding, unbinding, and then these stable localizations of Recce that land up towards the edge of the filament. And they come later in the movie. So you have a filament that builds, Recce comes and binds to it, and then over time, it just ends up at the end of the filament, right? So it's landing up at the lagging edge of the filament. Okay, and so now we can try to build a model with these information. We can build a model, which took off too quickly. We can build a model where you have this Recce filament, Recce binds to it, and we know that these bindings happen across the entire filament. But then you have these stable localizations that turn up towards the end of the filament. And these stable localizations remain even when there are bent filaments towards the poles, and they remain again at the lagging edge of the filament. And so we think what's going on is that Recce is binding to the Recce filament and sort of hitching on to it on one end and holding on to double-stranded DNA at the other end, and then walking. It's walking the filament on the double-stranded DNA. And it does this walk a few times. Maybe there is this event of searching also. We don't know. This is totally hypothetical. But we think there is walking. And then there is unbinding after that. And so this is what our working hypothesis is, that there's a Recce molecule. It binds, walks, moves the filament, and then unbinds. And then it goes through this process again. And this whole thing is driven by its ATPase cycle. Because Recce has ATPase activity. You said I have 5 to 10 minutes, right? So I'll take 1 more minute,
Yes.
and I'm done.
like the persistence and physical properties of the whole field?
Yeah, so I think I don't know the answer. I don't know the answer. It could be either we don't see sharp angles where Reckon is bound, and also given that there are the binding is transient, my guess is it's changing persistence length as it's moving the filament. That's my guess, but I don't have a good answer. This bridging, by the way, comes from in vitro data. There is published evidence to suggest Reckon can bridge the filament and double-stranded DNA. Okay, and finally, we have our own evidences to show you that this ATPase cycle does follow this model that I just showed you. And if you want to know more about it, we can talk later. Okay, and then finally, I'm just going to quickly acknowledge these guys. All of this has a polymer model associated with it that can recapitulate all of the dynamics we've observed. And this was done by our collaborators at IMBA, Ankit, and Anton's student, Ankit, and Anton did this work. And they have suggested that maybe this can explain all of the motion similar to what I told you. It's this paddling behavior. Reckon binds to the Recce filament, and it uses, instead of water, it's using the double-stranded DNA, the chromosome, as a paddle to push the Recce filament forward as it itself has a backward motion with ATP. And that can allow it to actually end up at the edge of the Recce filament at the end of one cycle of rowing. And I won't show you the model because this time not there. All right. Okay. And then all of this holds in a eukaryotic system also, so it's not a peculiarity of one organism we have studied. This seems to hold across at least 2 to 3 unrelated systems, which suggests that this ability to move around in the cell with a mobile and dynamic filament allows you to do repair in a more efficient manner. And this is our working model right now of how this occurs. Okay, with this, I'll stop. I have acknowledged already the people who have done the work and funding. Thank you for your attention.
So compare that in human beings, like something like a big fraction of DNA, you don't know what it codes for, it doesn't seem to that. Does this repair work the same way if it is in that non-coding part as well as the coding part?
Yeah, so I mean, at the level of sequence search, it should be the same. But in terms of coding, non-coding, it's interesting because in our case, we're seeing this effect of transcription on repair times. It appears that when we're hitting a fast transcribing region, which should be likely then coding, you get faster repair, maybe because the DNA is open and so you search more. And maybe you don't see that in non-coding regions. That's one possibility. But
there are no.
It's all coding, but there's variability in the levels of transcription across these regions. There's this one place that has super high transcription, which is orders of magnitude higher than the rest of the chromosome.
From the information theory point of view, if you want to define something like a Kolmogorov complexity or a Shannon entropy to a strand of DNA, I mean, if it's just the same sequence repeating again, be low complexity. But do you know if these sort of processes correlate with something like that?
It's a great question. I mean, it would be very nice to see whether something about the inherent properties of the DNA itself can also have either guide search or have an impact on search. And we didn't come at it from the physical perspective. We came at it via making break sites at various locations to try and get to what you are asking. But it would be really exciting if we could maybe build a model of whether the properties of DNA themselves may guide search. Yeah.
Something about the filaments coalescing together. Did you find any difference in velocities when there were more number of breaks with less number of breaks? Was it different? Because since they are coalescing together, there might be some cost of which helps them guide.
Amazing question. I don't have an answer to that. We should go back and analyze these data. That's a great question. I don't know. Yeah, I will get back to you. Thank you.
I make it say it's my ignorance, but so you have this situation where you find a particle create two chromosomes. So in a bacteria where you just have how does this system work? It looks for exactly identical sequence, so it works with somehow closely resembling sequence.
Yeah, so I mean, there is.
don't really have knowledge regions, why would the system evolve to begin with? What is the more fundamental function?
Really glad you asked that second question. So the first one, there is the possibility of homologous recombination. In bacteria, it's a rarer event because any gene you hit is essential for some, like there's more essential genes than non-essential genes. And so overall, you don't tend to see a high probability of homologous recombination. Tend to see it a lot in yeast. Again, mammalian cells, this rate drops quite a bit. So it is possible to do this type of recombination, which is a bit more erroneous. But also in eukaryotic systems, non-homologous end joining kicks in. So you wouldn't get recombination active all the time. And in cases where you may have a recombination template far, non-homologous end joining may actually be the preferred pathway. In bacteria, non-homologous end joining is a highly sporadic pathway. It's non-conserved. It's conserved in, I think, 6% of sequence genomes. So this is the predominant repair pathway. So bacteria do rely on this. Now, why would you do this if it's possible that more often than not, you wouldn't have the template? And I think it answers the first question also. We've been considering that maybe the reason to move is not for recombination in this manner, but it is to enable horizontal gene transfer. Because when you have incoming DNA, it can come from any location inside the cell. And Recce is going to coat that DNA, and then it needs to do the search. And so if you have now the picture of AGT instead of this recombination, then suddenly this motion makes a lot more sense because you would need to search. It's not a site-specific break anymore. And it's also not a perfect homology that you need to recombine. So searching gives you the chance of finding across the sequence space the best possible match to insert the DNA. And that was also perhaps how bacteria rely on recombination a lot more because rates of horizontal gene transfer in natural environments are a lot higher. And recombination is a very important pathway in natural bacteria. Exactly. Yeah, I mean, we've been curious to see whether we can track so we have this process where we can infect cells with phages and then track the phage DNA as it's trying to recombine. We want to try and see whether this pathway would kick in there and we'd be able to see it, in these swarmer cells with only one chromosome.
You mentioned that when there are multiple breaks, the filaments sort of coalesce and move together as one. If there are multiple breaks, they'll be carrying multiple open ends. Wouldn't that increase the chance of more error-prone rejoining and replication?
Yeah.
We need only 8 base pairs to commit to a replication, right? It's a repair in this case. So it's just not searching for just 1 or 2 fragments, but searching multiple fragments in a certain place. Chances would increase if there's a mismatch in 1 or 2 sites,
but each molecule is searching independently and it's multiplex. So the one thing we have to remember is that in the filament, it's not a full filament that searches at any given point. It's possible that only a limited number of molecules actually engage in search at any given point. And so when you have multiple break ends coalesced, is essentially just a multiplex search. So you may actually even reduce errors. So the chance of genetic rearrangements become high if you have break ends that are moving randomly in space, because then they can just collide somewhere, promiscuously and then do recombination. But here, if you have it more organized, my guess is it may reduce the chances of erroneous recombination. That's my guess. But again, I don't know the answer.
Just to is it looking at the first 8 or any random 8 in
any 8 within the filament? It doesn't have to be from the first molecule till the third molecule.
So the vacuum it's using their the ability was again into the search, and you said vacuum is associated with vacuum. Then I am not able to visualize how is that vacuum using DNA to that.
Yes, exactly why we drew this thing that has no support at all as yet. But we propose the following, that there's 2 motions, right? There's this movement, but then there are also searchings. we're hoping that we will find at some point a mutant that can move, of this complex that can move, but cannot search. And that way we can establish that movement may occur via this bridging and walking. And then searching is an independent thing that the filament can do as it's being moved around, right? And then that searching is not really dependent on RecN. That searching is dependent just on the property of the RecA, which is why search times don't change. But the movement is dependent on RecN. And so that's how we're reconciling it. It's only a hand wavy model. It doesn't answer your question. And I think it's a really, really important question for us to also understand. Where do you position the search itself in this process? No, but I mean, this is our working hypothesis.
If you can create these breaks in certain places, and then you make multiple breaks at the same time at particular spots, and then you accurately visualize how these breaks come together, how they fall off as per the location of the break. That would be.
Yeah. I mean, So I mean, so we've actually not done that experiment. It's a good one. Instead of making multiple single position breaks across many chromosomes, you make multiple clustered breaks on one chromosome itself and see how they are. It's a bit of a challenge. So one of the things you have to realize in bacteria is that you get this, where every time you do a duplication, right? So now when we have these two chromosomes, you got one chromosome here, one chromosome here. There's chance of breaking this, there's a chance of breaking this because this is an exact match, or there's a chance of breaking both. And we have a way to segregate them. But when we start to do this that you're asking, multiple breaks, the precision becomes harder for us to have.
Okay, I think with that, let's close the session and let's thank Anjana again for the Thank you.
Thank you.
Please join us in the West Campaign and you get to interact with Anjana more.